हेलो फ्रेंड्स, वेलकम बैक टू माय यूट्यूब चैनल। इस वीडियो में हम एमएससी जूलॉजी फोर्थ सेमेस्टर सब्जेक्ट बायोकेमिस्ट्री का टॉपिक माइटोकॉन्ड्रियल इलेक्ट्रॉन ट्रांसपोर्ट चेन का स्टडी करेंगे, जिसका इंट्रोडक्शन है। द माइटोकॉन्ड्रियल इलेक्ट्रॉन ट्रांसपोर्ट चेन इज ए फंडामेंटल बायोकेमिकल प्रोसेस that occurs in the mitochondria, often referred to as the powerhouse of the cell. This system is essential for energy production in eukaryotic organisms. The ETC forms the final stage of cellular respiration, where highly energy electrons are transferred through a series of protein complexes to generate adenosine triphosphate, the energy currency of the cell. Understanding the ETC is crucial for students in zoology and biochemistry, as it ties together concepts of molecular biology, bioenergetics, and physiology. Definition: the mitochondrial electron transport chain is a series of protein complexes and small molecules embedded in the inner mitochondrial membranes that facilitates the transfer of electrons from electron donors such as NADH and FADH to the final electron acceptor, oxygen. This process is coupled with the generation of proton gradient that drives ATP synthesis. Structure of the electron transport chain. The ETC consists of the following components. First, complex 1, NADH ubiquinone oxidoreductase. This complex accepts the electron from NADH, which are transferred to ubiquinone coenzyme Q. It pumps proton into the intermembrane space, creating a proton gradient. Second, complex 2, succinate dehydrogenase. Unlike complex 1, this complex does not pump protons. It accepts electrons from FADH generated in the citric acid cycle and transfers them to ubiquinone. Third, ubiquinone coenzyme Q, a mobile electron carrier that transports electron from complexes 1, 2 and 2 to complex 3. Complex 3, cytochrome BC complex. Electrons from ubiquinone are transferred to cytochrome C, a small protein that shuttles electron to complex 4. This complex also contributes to the proton gradient. Cytochrome C, a small soluble protein that transfers electron between complex 3 and complex 4. Complex 4, cytochrome C oxidase. These complexes transfer electron from cytochrome C to molecular oxygen, reducing it to water. It also pumps proton to strengthen the proton gradient. Mechanism of electron transport and ATP synthesis. The electron transport chain operates through a series of redox reactions where electrons are transferred from donors to acceptors. This reaction releases energy, which is used to pump proton across the inner mitochondrial membrane into the intermembrane space. This creates an electrochemical proton gradient, also known as proton motive force. ATP synthesis is driven by this proton motive force through the action of ATP synthesis, an enzyme complexes located in the inner mitochondrial membrane. Proton flow back into the mitochondrial matrix through ATP synthesis, which utilizes the energy to convert adenosine diphosphate ADP and inorganic phosphate into ATP. Key features of the electron transport chain. First, proton gradient formation. Proton pumping by complexes 1, 3 and 4 establishes a gradient essential for ATP production. Second, oxidative phosphorylation. The ETC is coupled with oxidative phosphorylation, where oxygen acts as the final electron acceptor. Third, efficiency. Approximately 36 to 38 ATP molecules are generated from the complete oxidations of one glucose molecule through cellular respiration, with the ETC playing a central role. Fourth, thermal regulations. The electron transport chain contributes to heat generation, especially in brown adipose tissue, through uncoupling proteins. Significance of ETC. First, energy production. The ETC is the primary source of ATP in anaerobic organism, fueling cellular processes. Second, metabolic integrations. It integrates with other metabolic pathways including glycolysis and the citric acid cycle. Third, regulations of reactive oxygen species. While the ETC generates ROS as byproducts, these molecules play roles in signaling and defense when tightly regulated. Disorder related to the ETC. Defects in the ETC can lead to severe mitochondrial disease including first, like syndrome, a neurodegenerative disorder caused by mutation affecting ETC complexes. Second, mitochondrial myopathies, muscle disease characterized by impaired energy production. Third, Parkinson's disease, associated with complex 1 dysfunctions and oxidative stress. Applications and research. Therapeutic targets. ETC components are targets from drugs in cancer, neurodegenerative disease, and metabolic disorders. Second, bioenergetics studies. Research on ETC elucidates mechanism of energy metabolism and mitochondrial dynamics. Third, evolutionary insights. Comparative studies of the ETC across species provide insight into evolutionary adaptations. Conclusion. The mitochondrial electron transport chain is a cornerstone of cellular energy metabolism. It not only powers the cell but also integrates metabolic pathways and regulates cellular homeostasis. Understanding the ETC is essential for addressing fundamental questions in biology and for developing treatment for mitochondrial disorders. References.